Hello everybody, welcome to this slideshow presentation. I will talk about excited states and complex environments with the use of polarizable force fields. I will present some of the results taken from my PhD thesis, and in particular these results are taken from the second part of the work. So in this slideshow I will start describing which are the prototypical systems that we take under exam in the kind of studies that I did, and what we mean by complex environment. So the typical system is built by a chromophore, so a molecule that absorbs light and gets excited to one excited state. This molecule is also called antenna, and usually in biological systems these antennae have a size of about 50-60 atoms. Around the antenna we can find an environment such as a solution, or even a more complex environment such as a protein matrix, where we have order of magnitudes more of atoms. In these systems we have common features such as a strong coupling between the antenna and the surrounding environment, and we have a large number of specific interactions between the atoms of the antenna and the atoms of the environment, and the computational goal is to provide an accurate description of the excited states of this embedded antenna in its environment. So the question can be at this point, why should we bother about excited states? Well, first of all, because one molecule, when it gets excited from the ground to excited state, can show new molecular properties that do not appear in the ground state normally. And it is also important to worry about excited states because many biological events can be triggered by the photoexcitation. So we can just think to the event of vision in human eyes, or the process of photosynthesis in the plants. From a more technical point of view, we have to remember that the modeling of excited states introduces sometimes some complications with respect to the treatment of the ground state, and generally sometimes there is less experience about the possible results of a calculation carried out in excited state with respect to the ones performed in ground states. So what is possible to do at the computational level to provide an accurate description of such a complex system? Usually what has been done for many years is to provide a partitioning of the full system and give a different treatment to the antenna and to the environment. Since the antenna is the molecule responsible for the transition from ground to excited state, it must be described with some quantum methods. So using quantum physics for this part of the system is essential, and it is the only possible choice. But the environment can be treated at a cheaper computational level because, of course, it is not possible to include everything in the quantum region. So we can use classical physics, which is more cost effective to represent the environment. So in the standard approach, the real atoms of the environment are replaced with fixed point charges taken from a force field. In the case of a solution, therefore, like a water solution, we will have that the water molecules of the environment are replaced by a set of point charges, one representing the oxygen and the other two representing the two hydrogen atoms for each water molecule. And at this point we will have a distribution of charges around the antenna that generates a standard a static electric field, and the presence of this electric field around the antenna should simulate the presence of the real environment, or at least give a first approximation of the real environment. So at this point we have the question if these classical charges are good enough to represent a real environment when we talk about excited states. And in this case, we can take the example of the light absorption of two small chromophores that we placed in a water environment. So in this case, we have a very reduced model of solution. We don't aim to reproduce any experimental value. We just want to compare how works the QMMM approximation with some reference number that we computed for the excitation. We can see from these numbers that the errors in computing the excitation within the QMMM framework are not negligible and range between 0.2 to 0.3 electron volts for these simple chromophores. Even if we go to a bigger system, such as retinal in rhodopsin, and we would try to study the light absorption, in this case representing the protein matrix with the QMMM, give us not negligible errors again. In these numbers, we can see that several different quantum methods chosen to treat the antenna are all at the same time far from the experimental value of 2.49 electron volt for this absorption. So there is probably something that is going wrong with our description of the environment, and our aim is to try to understand what we can do to improve to improve on these errors. So, sticking to the realistic case of the light absorption of retinal in rhodopsin, one thing that we can do is to try to expand the quantum region around the chromophore using a cheap computational approach, which is time-dependent DFT. So in this case, we want to try to see if we actually improve the environment going from a region, a small quantum region limited to the chromophore surrounded by point charges, to a more broad quantum region, all treated at the time-dependent density functional theory level. And actually what we see is that there is a clear shift in the excitation. We go from a range of 2.7 eV roughly to more or less 2.5 to 2.6 electron volt. So we are improving the description of the environment, giving a fully quantum description of a reduced cluster. Still, we are not, of course, going exactly on top of the experiment for several different reasons, but we see a trend at least that tells us that improving the environment brings down the excitation in this system. But the problem is that these kind of calculations are too expensive from the computational point of view, and most of all, we have to be limited in the use of time-dependent DFT because we are treating about 250 atoms, and it is not possible to use any other method for computational reasons. So we have to find a cheaper solution, and we want possibly to keep the description of the environment still classical, and at the same time we want to improve on the point charges description of classical QMMM. One idea is to use polarizable dipoles on top of the static point charges. So in this point, we do not describe the atoms of the environment only as a set of fixed point charges, but we assigned to each of them also an atomic polarizability. With this polarizability, it will be possible to compute an induced dipole moment on each classical site, and this will have several advantages. This way of modeling the environment is computationally much cheaper with respect to the use of an extended quantum region. The computational time that a typical calculation using polarizable dipoles is compatible to the typical QMMM calculations, but at the same time it brings a more realistic description of the coupling between the antenna and the environment, and this comes from 2 points. On one end, we improve the description of the ground state with the use of the dipoles, and at the same time we can allow the environment to respond to the excitation of the antenna, and this is the real important advantage that we have when we treat excited states, because with the standard QMM, the electric field that perturbs the ground state is the one that perturbs the excited state, and there is no environmental relaxation at all, while I will show that the environmental relaxation is crucial to obtain accuracy, and it can be done using polarizable dipoles. From the mathematical point of view, the introduction of dipoles introduces a complication, because working out the equations, we end up with a nonlinear Schrödinger equation, such as the one written in equation 1. So in this equation we have a static Hamiltonian H0, which is the same Hamiltonian that we would write for the isolated chromophore, and we have a potential term that includes, among other terms, the term of interaction between the classical dipoles, so mu A, where A is a label running on all the classical sites, and the electric field of the solute. Why it is a nonlinear problem? Because the dipoles mu depend on the wave function, because we write the induced dipoles in linear approximation as a proportion between the atomic polarizability and the total electric field generated by the in the system. If we put in a more explicit way the equation for each atomic dipole mu alpha, mu A, we have, as I said, that it depends from the electric field of the solute, and each dipole is also induced by all the other dipoles of the system. So we have here clearly a problem of self-consistency between equation 1 and equation 2, and we have to proceed doing self-consistency cycles, and this eventually leads to the generation of state-specific dipoles and state-specific potential. So in practice, we start from a certain wave function, we induce we induce the dipoles from this wave function density. These dipoles generate an external potential, and from on this external potential we re-optimize the wave function until the cycle is brought to self-consistency. So how can we compute in this way excitation energies, and how many routes we have to compute excitation energies using polarizable dipoles? Once we bring to self-consistency, so the two equations that I'm showing at the top of this slide, we can follow different paths. So as I said, once we solve the nonlinear Schrödinger equation, we end up with a set of different potentials and different optimal dipoles for the different states of our problem. One way to compute an excitation energy is, for instance, to polarize the environment only on the ground state. In this way, we only want to compute the dipoles in equilibrium with the ground state density and freeze this environment also for the excited state. In this case, we are only trying to improve the description of the ground state, but we do not have, again, any response from the environment with respect to the excited state density. So we are in a situation somehow similar to the one of the classical point charges embedding, and the difference is only that we are improving the description of the ground state, which still can give us some insights on the physics of our problem. A second step is instead to use state-specific dipoles and the corresponding energies, so compute so-called state-specific excitation, where the energy of the excited state is computed in the potential generated by the optimal dipoles for the excited state electronic distribution, and the excitation energy is computed taking the difference of this quantity with the energy of the ground state computed in the presence of the optimal ground state dipoles. If we use this equation to compute an excitation energy, we recover the so-called differential polarization effects that have a big role when the electronic distribution of the excited state is significantly different from the electronic distribution of the ground state. So let's see an example of how this polarization works. Here we are describing the excitation of MCP, this green molecule that we see on the left, in water. This excitation is characterized by a HOMO-LUMO transition, and it is linked to a blue shift when we are in a polar environment such as the water solution. On the plot, we can see the trend of the excitations in different embedding conditions. At the extreme left of the plot, we have the isolated molecule, and we see, as expected, that when we describe the environment with classical point charges, we have a very strong blue shift. We are trying to compare our numbers with respect to a calculation where we use the same quantum method for all the molecules of the system. So the reference is found at the extreme right of this plot, and it is given by a calculation where all the water molecules and the MCP molecule are treated at the same time with the three quantum methods that I'm showing in the caption of the plot. As I said, the excitation energy computed using classical point charges is quite has a certain error with respect to the reference values that we are trying to improve using polarizable dipoles. If we compute the excitation energy polarizing the system only with respect to the ground state, so in the Pol-GS condition, we see a further enhancement of the ground state, and this is a consequence of the use of a stabilization of the ground state, while the excited state does not respond to the excitation. So the gap opens even more with respect to the embedding condition given by the classical point charges. But we can see that we recover a lot of this blue shift, and we go somehow towards a better agreement with the fully quantistic reference number when we introduce the state-specific polarization of the environment. So when we allow the dipoles of the environment to relax on the excited state density of the molecule. So this state-specific correction, as I said, is very important when the electronic distribution of the excited state is very different from the one of the ground state, and deriving the equations and following a perturbative approach, we can find that the correction of the state-specific embedding with respect to the ground state polarized environment is proportional to delta mu, where delta mu is the difference between the excited state dipole moment and the ground state dipole moment. So we have this nice and interesting result for a model system, and we want to try to use this tool of polarizable force field to describe a more challenging problem, such as the light absorption of retinal in rhodopsin. Rhodopsin is a very debated protein. There are many things that are known for sure, and some other things that are not known. So among the things that we know are listed here in this slide. So the excitation of retinal in rhodopsin is an excitation characterized by charge transfer with a migration of electron density from the beta ion ring on the left on this figure to the terminal of the chain that is linked to the lysine residue on the right of the figure. It is a pi-by-star transition associated to a high value of the oscillator strength, and the migration of the charge generates a repulsive interaction with the residue of glutamic acid close to the chromophore, and this generates a blue shift with respect to the absorption of the isolated antenna. The role of the rest of the protein is to create a quenching of this blue shift, eventually giving the observed experimental absorption. There are, anyway, things that are not clear, and in particular, it is not clear which is the exact magnitude of this quenching. Some authors say it plays a big role, some other authors say that it plays a negligible role, and Again, it is not clear which is the exact nature of the interactions between the environment and the chromophore, and ultimately we have to question if a purely electrostatic modeling of these interactions, so if using a standard QMM, is enough in these cases. The picture that I introduced before was suggesting us that there were some limitations in the use of classical point charges and the non-polarizable force field because a bunch of quantum chemical methods were predicting absorption energies all internally consistent but far from the experimental value. So at this point we want to see if using a state-specific polarization of the environment actually can help us, and to do so we have anyway to play it smart, and we want to introduce the polarizable force field in an egg-board around the antenna, and we select a radius of about 15 angstrom around the chromophore, while all the other sites are still treated with static point charges. So here we go with the results. We see that, of course, we have a trend of blue shift, as I said, going from the isolated chromophore to the classical point charges, and the PolGS produces a slightly enhancement, similarly to what we see in the case of MCP in water, of these excitations, but the real surprise is that the state-specific correction doesn't help us in this case as it was doing before. In this plot, I am showing three different trajectories extracted from QMMM molecular dynamics, and I'm also showing an yield structure. So what we see here is that PolSS brings a very small correction, and these numbers are all computed using time-dependent DFT and the Cambit-Relip functional. So what we can do at this point, actually we are not out of resources, because using the polarizable dipoles we can do we can follow another route to polarize the environment, and in this case we can try to give a linear response solution of the nonlinear Schrödinger equation using this, what I will call linear response polarization. Before describing it, I will show how it behaves in the case of retinal rhodopsin, and then I will show some mathematical background of it. So when we introduce this linear response polarization, also called resonant polarization, we see that the correction with respect to the PolGS embedding condition is very big, and eventually if we combine this resonant polarization or linear response polarization with the state-specific correction, we achieve actually overall a good description of the excitation, which is in fair agreement with the experiment. So what are we doing when we follow the resonant or linear response polarization? In this case, we are simulating actually different kind of interactions with respect to the ones that we were describing using state-specific polarization. Just to make a summary, I said that we can compute classical dipoles and put them in equilibrium only with the ground state, or we can recover a state-specific correction accounting for an electrostatic relaxation of the environment on the excited state density of the chromophore, and this correction is induced by the difference between the excited state molecular dipole moment and the ground state molecular dipole moment. In the case of retinal rhodopsin, this quantity, delta mu, is supposed to be large, so we are quite surprised to see that actually we are not observing the expected large correction of PolSS with respect to PolGS. This kind of polarization recovers the classical electrostatic and the inductive coupling terms between the chromophore and its environment, and now with the linear response or resonant polarization, we are inducing the dipoles not on the density of the ground state, nor with respect to the density of the excited state, but we are inducing them with respect to the transition density associated to the electronic excitation. In this embedding condition, the response of the environment is not induced by the difference between the excited state molecular dipoles or the ground state molecular dipoles, but by the transition dipole moment associated to this excitation, which is an intrinsically quantistic quantity. So, following some derivations showed in reference papers and following the interpretation of several authors, we can say that this kind of interaction, these resonant interactions, are non-classical and sometimes are considered as dispersion terms in the coupling of the chromophore with its environment. If we follow a perturbative approach, we can see that while the state-specific correction, as I said, is induced by the delta mu, and the resonant correction is induced by the transition dipole moment, we can have that the two corrections relate to each other in the way that I'm showing here in this equation at the bottom of the slide, and more details can be found in this reference paper. So at this point, what have we learned from this, and what can we do? So we had the surprise that accounting for the electrostatic terms is not enough in the case of rhodopsin, even if we are talking about a charge transfer excitation. This tells us that the coupling terms goes beyond the electrostatic picture, and we need to include another kind of polarization that takes into account non-classical effects, and this can be done using the resonant polarization of the environment. Of course, the ultimate clean solution is still to include as many atoms as possible in the quantum region to create a large cluster and to compute very accurately the excitation energy, but a very good and fast solution, still maintaining a classical description of the environment, is given by the combination of the correction given by the resonant polarization of the environment with the state-specific polarization. Moreover, the use of these techniques allow us to obtain additional insights from our system, and actually we can fingerprint the response of the single amino acids of the protein to see which of them responds the most. In this plot, I'm showing the response of the state-specific level in the upper panel with the red lines, and the resonant polarization in the lower panel with the green lights. So we can see that basically the same set of amino acids responds in the excitation, but of course the magnitude of the polarization energy is different in the two approaches. So a useful thing that we can do with this information is to create a cluster with these amino acids that are important in the excitation and try to compare a calculation at the cluster level with the calculation using the polarizable force field. So we create a cluster with these amino acids, and we compare in this plot how an extended quantum calculation on these amino acids compare with quantum calculations only on the chromophore that is surrounded by the same amino acids, this time described with polarizable force field. We can see that the error is quite reasonable, so we have a shift that is about 0.05 electron volt, and is more or less the same error that we obtain when we just describe the ionic pair of the chromophore and the glutamic acid. So we have good reasons to think that also in the description of the full protein we are not far from the results that we would get with a very extended quantum calculation that anyway is not possible to perform. So to give a summary and to conclude this slideshow, we have seen that the improved description of the environment translates directly in an improved description of the excitation properties of a chromophore, and a very nice way to improve the description of the environment is to use polarizable dipoles, which are computational cheap and tells us a lot about the physics of the system under analysis. And in doing this, we have to be aware that different polarization schemes rely on different physical and rely on different physical equations and simulate different effects, and therefore they can lead to very different results. And finally, for the case of rhodopsin, we have learned that electrostatic embedding doesn't tell all the story, and we can do much better using polarizable dipoles, and we have to combine resonant polarization and state-specific polarization. But if we really want to have a clean result, we have to use an extended wave function method, or anyway a quantum method that includes as many atoms as possible, also from the environment. But this introduces some complications and poses some questions. For instance, which wave function should we use to describe such a large system, and at which computational cost do we want to go to be satisfied by the accuracy of our results. So to conclude, I want to thank all the collaborators that worked on this project, and of course to you for your attention.